This is a production of Cornell University.
Thanks everyone for being here. So I'm a master's PhD student, which means that this presentation is going to have elements of both what I've done in my master's as well as what I plan on doing in my PhD. And so really this presentation is a broad encapsulation of my entire graduate school experience. And I think that when designing a presentation that's so broad, it helps by starting with what drives my research interests. And the most important moment in me as an agroecologist was my high school senior project, where on a whim, I decided to study the importance of biodiversity in natural and managed systems. And so the next two slides are going to be slides from that presentation. So I didn't really know much about biodiversity, but I realized that I wanted to have a project which was on how different people interacted with wilderness, agriculture, and urban areas, and how biodiversity influenced these interactions. And I also realized that to learn more about biodiversity and answer my questions, I wanted to break up my project into 4 questions. So I started by asking, what is biodiversity and what are ecosystem services? And second, what's the importance of an ecosystem service? Third, what are the current trends in biodiversity and why should we care? And then finally, how can we promote biodiversity? And I had a great time asking myself these 4 questions. And so when I finished high school, I decided to study agriculture. And I chose to study agriculture because I learned that agriculture covers about 1/3 of the world's land area. And 66% of agricultural land is dedicated to 5 crops. So as someone who was interested in promoting biodiversity, I realized that agriculture has, or at least conventional agriculture has a tremendous homogenization effect on biodiversity. And I also learned that a tremendous amount of inputs go into maintaining conventional agricultural systems. So there's billions of pounds of pesticides that are used annually, and there are consequences to this over-reliance on pesticides, including non-target effects on biodiversity, as well as effects on human health. Across the world, the main type of pesticide used are herbicides. And so herbicides are a class of pesticide that is used to manage plants or weeds. And the consequence of over-reliance on herbicides has been the increase in cases in herbicide resistance. And since the 1980s, we've had a pretty consistent and steep increase in weeds that are not as responsive or not responsive at all to herbicide treatments. And the increase in herbicide resistance is particularly problematic when taken into account with the fact that the agrochemical sector has not put out a new herbicide mode of action for the last 30 years. So quite literally, farmers can't stay ahead of the herbicide resistance curve. And in New York, herbicide resistance is a real issue. There are 4 weeds which are officially defined as herbicide resistant, and they're common groundsel, smooth pigweed, common ragweed, and common lamb's quarters. However, there is reported herbicide resistance in horseweed, tallwater hemp, and Palmer amaranth. And these are pretty common weeds in New York. And so the fact that there's herbicide resistance in these species shows that it's a real problem, and it's a real problem here in our state. On the flip side of the coin, a lot of weed management in farm systems, and especially in organic systems, is accomplished through tillage. However, overuse of tillage drives erosion. In fact, most erosion occurs on croplands and is due to tillage. And erosion is quite problematic to the sustainability of agriculture, as about 1/3 of the Earth's arable land has been lost to erosion in the last 40 years. Okay, and so compared to the average natural area, erosion rates in agricultural settings are 1 to 2 orders of magnitude higher. And so that's displayed in this figure here, where the x-axis shows erosion rates. This band here is what's defined as the acceptable rate by the USDA, and we have agriculture compared to 3 natural settings. And we can see that 1, agriculture has pretty high erosion rates, and that 2, the median rate of erosion in agriculture is higher than what's acceptable by the USDA. And so in response, with a lot of the problems with conventional weed management, 2 agroecologists, Matt Liebman and Eric Gallandt, proposed the many little hammers paradigm of weed control. And the idea behind this approach is to combine tactics which are weakly effective when applied in isolation, but can provide effective weed suppression. And by spreading the burden of weed management across several methods, a farmer can promote farm system resilience because their weed management program isn't contingent upon the success of one big hammer of weed control, like tillage or herbicides. Likewise, because each of these management practices are relatively weak in isolation, there is a reduced potential for the development of weed resistance because there is a less intense selective pressure on a weed species. Okay, so the many little hammers approach can be more sustainable in the long term. And another interesting emphasis of this approach is that diverse weed management practices can be synergistic, which means that when a lot of weed management practices are applied together, their effect on the weed community may not necessarily be additive. In fact, it can be multiplicative or have interesting interactions. And the use of ecological knowledge to manage weeds is called ecological weed management. And there are various mechanisms by which ecological management can reduce yield loss. And these are described by Bastians, and he has 4 graphs where yield loss is on the y-axis, the density of weeds is on the primary x-axis, and the number of weed seeds in the seed bank is on the secondary x-axis. And the first mechanism of weed control is reactive weed control. And this is quite a conventional method of managing weeds. It is based upon killing weed plants that have emerged. And so practices like secondary tillage and hand weeding kill weeds that have emerged to reduce their density and then reduce yield loss, okay? A mechanism of ecological weed management is the reduction in the emergence of weeds from the seed bank. And so practices like mulching can reduce weed seedling emergence rates, okay? The third, or another mechanism of ecological weed management is reduced weed crop competition. And unlike the other 2 mechanisms, the idea here is not to reduce yield loss by reducing the density of weeds, rather to reduce yield loss by increasing crop tolerance to any given density of weeds. And there are various crop management practices which can accomplish this mechanism of ecological weed management. For instance, fertilization can be modified to change weed crop competition dynamics. So a farmer can band their fertility across a crop row to prioritize fertilizing the crop instead of the weed, as they would if they were broadcast fertilizing. a farmer can also select for more competitive cultivar, or they can do intercropping. And intercropping is the planting of more than one crop plant on any given area of land. And intercropping can affect resource availability dynamics and consequently affect weed crop competition. Another mechanism of ecological weed management is the reduction of seed recruitment into the seed bank. And so this is any practice that limits weed seed rain. So for instance, mowing before a weed sheds its seeds would accomplish the reduction of the weed seed bank. And so when I was learning about these practices of ecological weed management, I became particularly drawn to the use of intercropping as a mechanism to reduce weed crop competition. Because remember, my primary motive in research is to promote biodiversity in agricultural settings. And so I thought, wow, great. By having intercropping, we can reduce reliance on conventional weed management practices while also promoting biodiversity in agriculture. So when I started my master's, I knew that I wanted to look for a project in intercropping. And at this beginning phase of my graduate program, I went through a phase where I was consuming a lot of theoretical literature to create the underpinning for my master's. And I became very attached to niche partitioning theory. And niche partitioning theory says that plant community diversity enables the exploitation of more niches. Okay, and a niche is any range of environmental conditions in which an organism can live. And the way I think of niche partitioning theory is by thinking of siblings. So if you have two siblings which are very similar in age and share very similar interests, there might be more potential for them to compete. Conversely, if you have two siblings which differ in age and differ in interest, there's less potential for them to be more competitive. And the same idea applies to plant communities, where if you have a plant community of very similar plants, they're likely to exploit the same resources and be very competitive with one another. Conversely, if you have a lot of diversity in your plant community, especially functional diversity, your plants will be using different resources, and consequently, there is less potential for competition. The application of niche partitioning theory into agriculture has been done by many scientists, but I was particularly drawn to the resource pool diversity hypothesis. And this hypothesis was postulated by Rich Smith, who's on my committee, and Matt Ryan, who's my advisor. And the idea of the resource pool diversity hypothesis is that agricultural practices influence the extent to which niches are partitioned between crops and weeds. And that the more diverse a management system, the less potential there is for weed crop competition. So the idea is, if a farmer plants the same crop year in and year out, they're likely to select for a weed community that's very similar to their crop and consequently very competitive against their crop. Whereas if a farmer diversifies their management system through practices like crop rotation or intercropping or varying their fertility and tillage regimes, they will keep their weed and crop community out of sync, and consequently there will be less intense weed crop competition. And an interesting research study that's kind of related to resource pool diversity hypothesis was one that found that intercropping can reduce the total weed biomass, as well as influence soil nutrient dynamics relative to monocultures. Okay. Another interpretation or extension of niche partitioning theory that I was very interested in was the microbial mediated resource hypothesis. And this hypothesis says that rhizosphere soil microorganisms can mediate plant resource availability. And a rhizosphere soil microorganism is a microorganism that's associated to plant roots, okay? And so the idea here is that diverse plant microbe relationships can promote niche partitioning. And this research this hypothesis has interesting contextualization when taken into account with research that has shown that farm system management can affect rhizosphere microbe communities. And so that's displayed in these two plots. These two plots are non-metric multidimensional scaling plots, which means that they're ordinations. Okay, so each of these points shows an entire rhizospheric community. And A is bacterial rhizospheric communities, whereas B is fungal rhizospheric communities. So the greater the distance between points, the more different communities are, okay? And the color of these points denotes the site from which the soil was taken, whereas the shape of the points denotes farm management system. And the finding of this paper was that the primary method of separation between rhizospheric communities was the location of which they were sampled. However, there was a significant effect of farm management system on the rhizospheric microbial communities. And I think that's very well shown in soil fungal rhizospheric communities. And so at this site, we can see that there is a clear separation in the organic and conventional systems. So, given all this past work, when I came to the drawing board and decided to formulate my master's project, I decided to ask, do soil nutrients and microbes mediate intercrop diversity legacy effects on competition? And a key word in my question is that I'm looking at legacy effects. Okay, I'm looking at whether intercrop diversity has a rotational effect or a carryover effect. Because as I mentioned before, there's past research that has already shown that intercrop diversity can affect weed crop competition and soil nutrient dynamics, and that broadly, farm system management can affect soil rhizospheric microorganisms. So I wanted to see whether these effects had a legacy on weed crop competition. And given that my work was based off of niche partitioning theory, I decided that my hypothesis would be that weed crop competition would be greater in soils from lower compared with higher diversity treatments. Okay. So today, for this presentation, I'm going to share the results of an experiment which were recently published. And this was a greenhouse weed crop competition experiment. And I was very fortunate in that I was able to answer my questions of intercrop diversity legacy effects on weed crop competition by taking advantage of a field experiment that was managed by a grad student in my lab and by B. Finley from 2016 to 2019. And this experiment had 4 diversity treatments in annual and perennial forage systems. Okay. And this experiment was replicated in York, Vermont, and New Hampshire. And by different diversity treatments, I mean that there were different levels of diversity. Okay, so the low diversity treatment was a 1 variety, 1 species monoculture. There was a conspecific diversity treatment, which had 4 varieties of 1 species. A heterospecific treatment, which had 4 species, and each of these species, there was only 1 variety of this species. And then finally, there was a high diversity treatment, which had 4 species, and each of these species, there was 4 varieties of these species present, okay? And so one important note that I just wanna point out now, because it's gonna affect my interpretation of the results, is that notice that the treatments are all based in a crop. So the perennials are all based in alfalfa. So in the low and conspecific treatment, you only have alfalfa, whereas in the hetero and high specific treatments, you have alfalfa and the introduction of perennial grasses. And the same goes with triticale, and sorghum Sudan grass in the winter and summer annuals. Okay. So for the greenhouse experiment, I sampled soils from the New York site in 2018 during this diversity conditioning phase. And I tested weed crop competition in a greenhouse experiment, and I tested the effects of soil nutrients and microbes by having three greenhouse treatments. I had a field soil treatment, which was 50% biologically active field soil to 50% sterile potting mix. I had an inoculated treatment, which was 5% biologically active field soil and 95% sterile potting mix. And then I had an inert treatment, which was a 100% sterile potting mix control. And the idea here was that the field soil treatment would have both the nutrient and microbial legacies of the intercrop diversity treatments, whereas the inoculated soil treatment would attempt to minimize soil nutrient differences while maintaining unique microbial communities. And the reason I chose this ratio and the reason we believe that this is possible is that it follows the methodology of plant soil feedback experiments. And plant soil feedback experiments attempt to test the functional effect of soil microbes on plant growth. And they try to ideally minimize nutrient differences between soils. So it's a rough but a contemporary technique. So the way I tested weed crop competition was by growing a density gradient of weeds across the 4 diversity treatments. So I grew 0 to 6 common lamb's quarter weed plants and 1 sorghum Sudan grass plant in all of these pots. Okay, and so I had the 4 diversity conditioning treatments that were present in the field in annual and perennial systems. And then I had my different weed densities. And then I had this in the field and inoculated greenhouse treatments. Okay, the field treatment being soil nutrient and microbes, the inoculated soil treatment being minimizing soil nutrients and trying to maintain microbial effects. And then I had my inert control. And by seeing the reduction in the crop biomass as a function of increasing weed density in each of these diversity treatments, I was able to model crop weed competition through competition curves. Another thing I was able to do was by comparing the biomass of a crop in the inoculated to inert soil, I was able to quantify plant soil feedbacks. And plant soil feedbacks are the relationship between plants and their associated microbial communities. And so, for instance, this would be an example of a highly negative plant soil feedback, because the plant growing in the inoculated soil is much smaller than in the 100% sterile inert soil. Okay, and so in this case, this would indicate that this soil has a net pathogenic effect on crop growth. So this next slide is going to show you the results of my weed crop competition experiment. But before I do that, I want to dig into what composed my competition curves. So my competition curves were 2-parameter models. The first parameter was A0, and the A0 parameter basically reflects the y-intercept. And consequently, it shows yield potential, right? It shows how much the crop can yield in the absence of weeds. The IW parameter is the shape parameter of these curves. And consequently, it's the competition coefficient. Because the higher the IW term, the steeper this initial slope in the curve. And consequently, the more intense weed crop competition. Okay? And so, just so we clarify, crop biomass is on the y-axis, weed biomass will always be on the x-axis. Okay, and I compared curves through f-tests. So My F-tests will always be across diversity treatments. So remember, these are the 4 diversity treatments, high to low. And this is the field soil treatment of the greenhouse in the annual system. Okay, and so these letters denote pairwise comparisons, which will always be down the column across diversity treatments. So this is the full figure, all right? And it's a lot to digest. So if you focus in on the competition curves, which are statistically different, there are 2 interesting conclusions to this competition study. And they apply to the annual and perennial systems. So the first conclusion, in the annual system, we found that weed crop competition was stronger in soils with a greater crop diversity legacy. And so this is opposing my hypothesis. You know, we did see a diversity effect, but in these soils, it was the opposite effect than what we were expecting. And so let's dig into this. So these 2 curves are statistically different. And I feel confident in saying that competition was more intense in the heterospecific treatment than the conspecific treatment, because the IW parameter, which is this initial slope term, is higher in the high diversity treatment than the low diversity treatment. And the same goes for in the inoculated soil treatments. At the same time, the A0 parameter here is the same or very similar. So the conclusion here is that these curves are statistically different because of differences in weed crop competition intensity. Okay, so that's why I feel good about this first conclusion. In the perennial soils, the IW parameter is also higher in the high diversity treatment compared to the lower diversity treatments with statistically different curves. However, there is considerable variation in the A0 parameter, which is the y-intercept term. So here, the differences between curves may be driven by crop yield potential. So I can't confidently say that crop weed or weed crop competition was higher in the high diversity treatment. But if we look at crop yield potential, there is a second set of results which are really exciting and interesting. So this figure shows crop yield potential. So this is the yield of the greenhouse crop in the absence of weeds. Okay. The x-axis is the 4 diversity treatments, the annual and perennial systems. This is the field treatment in the greenhouse, so both nutrients and microbes. And this is the inoculated treatment. Okay, so this attempts to minimize nutrient differences while maintaining microbial effects. And we can see that, as I explained in the figure before, the annual system has no differences in yield potential. So that was the no differences in the A0 parameter. So that makes sense. But in the perennial system, we can see that there is an increase in crop yield potential with diversity legacy in the field treatment and a decrease in crop yield potential in across diversity treatments in the inoculated soil treatment. So this is a very interesting and exciting result, I think. And to contextualize this result, I think we can draw upon the biomass of crops that were growing during the conditioning phase right before I sampled my soil. So this figure here shows the biomass of grass in red bars and legume in blue bar crops right before I sampled my soil. And this figure shows plant soil feedback values. So notice that all of these values are negative, so it's basically how pathogenic soils were. And again, in the annual soils, there were no differences in the biomass of conditioning crops, and all of the conditioning crops were grasses. Likewise, there were no differences in plant soil feedback values. So again, crop yield potential in the annual system, no results. But if we focus on the perennial system, here the story gets really interesting. So let's explain what was going on in the conditioning phase. Remember how I told you that the perennials were based in alfalfa? So you can see that the low and conspecific treatments only have legume biomass. However, as the experimental design called for, there was an introduction of perennial grasses and the hetero and high specific treatments. Okay, and there's a significant decrease in legume biomass in the high diversity treatment compared to the low diversity treatment. And I think that these differences in the crops that were growing during the conditioning phase could very well explain the results of crop yield potential in the greenhouse experiment. So first, let's start with the inoculated soil treatment, where crop yield potential decreases with conditioning diversity. So remember, the inoculated soil treatment is attempting to look at only microbial effects. And there's research that shows that rotational diversity will have a large influence on the pathogen load of soils, or the plant soil feedbacks of these soils. Wherein if you rotate a grass system to a grass system, it will have much more negative plant soil feedbacks than if you rotate a grass system to a legume system, or a grass to a legume crop. And remember that the crop that I was growing in the greenhouse was a grass. So my interpretation of the decrease in crop yield potential with conditioning diversity is that the presence of a grass legacy right before we had sampled soils resulted in more pathogens in the hetero and high diversity treatments. And this is affirmed by my plant soil feedback study, which finds that there is a decrease in plant soil feedback value as a function of conditioning diversity, which is indicating that these microbes were having a greater net negative effect as a function of conditioning diversity. For the field soil treatments, okay, here are the soil treatments that had both nutrient and microbial legacies. I think that the way we managed our crops before soil sampling explain these results. Remember, the experiment is trying to mimic a forage crop system, okay? And so we were cutting and removing the herbage from the system multiple times a season. And there is research that shows that multiple cuts and removals of alfalfa herbage can reduce soil nitrogen availability. Whereas multiple cuts of perennial grasses can increase root exudation and increase nutrient cycling. So I think that the presence of a perennial grass legacy in the higher diversity treatments might have contributed to the higher crop yield potential in the field soil treatment. So the conclusions of this experiment that I did for my master's were that 1, crop diversity can have a legacy effect on weed crop competition, and that 2, soil microbe communities can mediate crop diversity legacy effects and perhaps have an independent and potentially opposing effect on crop yield potential compared to soil nutrient microbe and nutrient legacies. And so that was the figure that I just showed you with the opposing trends. So this was my master's, where I was looking at ecological weed management through intercropping, and there's multiple projects, but that was the underlying theme of my master's. And in my PhD, I'm going to continue to look at ecological weed management, but I'm looking at multiple mechanisms of ecological weed management. I'm not just looking at weed crop competition, and also all of my experiments are going to be focused on a reduced till organic system. And I'm looking at two pathways to reduce tillage. I'm looking at a roller crimped system, which is a system that rolls a cover crop with a very heavy roller to terminate the cover crop and create a mulch, and then seeds a cash crop directly into that rolled mulch. And I'm also looking at reducing tillage through perennial grains. So these are crops that stay in the soil for multiple years. And I have 4 big experiments planned for my PhD. The first experiment is looking at cereal rye and soybean-based population weed suppression. And the questions here are, well, how does the low nitrogen environment that's fostered by the decomposition of cereal rye when it's rolled in a mulch system affect weed suppression? And I also want to see whether light interception from an increased soybean density, I want to see whether that promotes weed suppression. And because I'm looking at 2 tactics here, I also want to see whether a low nitrogen environment and a high soybean seeding population have a synergistic effect on weed suppression. My second experiment is also in a roller crimped cereal rye soybean system, but here what I'm varying is the biomass of cereal rye. And I want to see how different biomass of cereal rye affects weed emergence from the seed bank. Another thing I'm doing in this project is I'm looking at the functional diversity of the weed community. So I want to see how different biomass treatments of cereal rye will affect weed functional traits. And then the third and perhaps biggest experiment of my PhD is a broad assessment of the compatibility of cover and cash crops for organic no-till. And so here I'm seeing a whole host of winter and summer cover crops and how they roll for winter and summer cash crops. And so there's various sub experiments or research priorities in this experiment. And so the first is just broadly seeing the weed suppressive abilities of several winter and summer cover crops compared to a tilt control. The second research priority is seeing how cash crops emerge in these rolled cover crop systems. And then the third and perhaps most theoretical question in this experiment is assessing how weed suppression is driven by the mulch of a rolled cover crop versus the presence of roots in this system. So remember that unlike a conventional annual system where there is primary tillage, maybe many times a season, and the rapid decomposition of cover crop roots, the idea here is that the cover crop roots stay undisturbed. So I want to see how the mulch versus roots of a rolled cover crop impact weed suppression. And finally, the last emphasis or last project in my PhD is testing for synergism between weed management tactics in establishment year Kernza. And Kernza is an emerging perennial grain crop with major corporate investments. So companies like Patagonia Provisioning and General Mills have invested in this grain. However, one of the main issues with Kernza is getting it to establish well. And within that establishment year, Kernza is a very poor competitor against weeds. So what I want to do is I want to see whether there's a positive interaction between various mechanical and biological weed management practices. So these are the 4 projects of my PhD. And today I've shared a lot. And first of all, I would like to thank Ms. Trinnell, who was that high school English teacher who gave me the intellectual space to do what I wanted. I think this is a testament, if anything, this project in my grad school is a testament for the importance of giving young people intellectual creativity. And I also want to thank Ann Bybee Finley, Chris Pelzer, Sandra Wayman, and all the research assistants and everyone associated to the lab for helping me create these ideas and get the work done. These people have been great help and great friends. So with that, I'm done. And yeah, I'll open it up for questions.
All right, nice job, Udi. Not sure I can see any other questions, but you can feel free to unmute yourself if you have a question. There's one question in the chat from Ruby. I could read that to you, Udi, or if you want to look at it yourself. It's basically asking about the connection and interaction with natural farming, both Korean and Japanese natural farming, and whether or not you had been thinking about that that type of approach. Ruby says that there's purposeful isolation of indigenous microorganism communities, which are diluted and applied to crop systems to provide inoculation of beneficial microbes. And she was wondering if you have any knowledge of people using these practices for weed management.
Yeah, so I'm not particularly knowledgeable about Korean or Japanese natural agricultural practices. So I don't know too much about those systems, but what I do know is that there is research looking at how inoculation with different microbial communities affects weed crop competition. And I am not looking at how, I've never done a project where I've purposely inoculated a cropping system with a different soil or microbial community. But I know people who do that. I think it is a very interesting and exciting experimental avenue. I think this is very, like, my personal opinion here. I think that I question how successful application of microbial inoculum is in a field setting where there's a whole host of microbial community already present, and which will compete with that microbial community. I wonder how establishment of that microbial community will work in a field versus greenhouse setting. So I think that's a potential limitation that I can imagine. But I do know people who research that and would probably know much more. So those are my opinions.
All right, so we have another question from chat, and Ben was asking how do you sterilize soil? How did you sterilize your soil, Udi?
Yeah, that's a great question, Ben. So a lot of thought went into that, and it's impossible to truly have a completely sterile solution. But my potting mix was a 60% vermiculite to 40% triple washed sand solution. So vermiculite and sand are both, firstly, fairly inert materials. Okay, so we weren't using some greenhouse potting mix, which has, you know, a lot of incorporation of matter. And then what we did is we autoclaved that potting mix. And we autoclaved it and then immediately used it in the greenhouse experiment. Of course, a greenhouse, by definition, is not a sterile environment. So there are system limitations, but all plant soil feedback, or many major plant soil feedback experiments are done in this greenhouse setting. And so the responses are at least strong enough to see differences in microbial functional effects on crop growth.
Yeah, thanks, Udi. So Jeff had a question. Jeff Liebert was asking if you could elaborate on what some practical applications might be from your master's research findings. So
yeah, so, and I also see that Brian Brown asked that question. And I think that's a really, really good question, right? Especially doing agroecology and you know, working with an applied system. I think a really interesting finding to my work is that while my hypothesis that diversity legacy effects would produce less intense weed crop competition wasn't upheld, I found that rotational diversity did affect crop weed competition, or at least it affected crop yield potential. So I think that the functional implications or applied implications of my work is that I would tell a farmer, look, like you can affect crop weed competition, at least crop weed competition curves through rotational diversity. So I would emphasize the importance of having a rotation that has diverse crops, right? To not rotate a grass to a grass or a legume to a legume. So I think that is an interesting implication of this master's work.
Thanks, Udi. There are a couple more questions here. I'm not sure if you could read them or if you'd like me to continue.
I can read them. So Andy asks, I mentioned characterizing changes in weed functional traits, and he was wondering how I would do this. So this is something that I'm really excited about, and I would be doing with Stéphane Cordeau in France. The idea here is to, there are databases that have descriptions of various functional traits in a weed community. So for instance, it describes like root length or root structure or average leaf size. And the idea here is that I would input all of these data and be able to create some sort of distance matrix between the weeds in my community. So it'd be very similar to the idea of non-metric multidimensional squaring on just the biomass of a community. But instead of looking at that, I'm looking at the like the relevance or the presence of certain functional traits, and then applying some sort of multivariate technique like a permanova to analyze whether there's significant differences. So Ashley asks what size pots I used. I think they were half-gallon pots. They were like, actually they're a little bigger. They're like this and like this. So that's the size I used, like half-gallon to 1 gallon for this master's experiment. When I was at Montana State, I used smaller pots. I probably used a 1/4 gallon pot. So there is variation in pot size for plant soil feedback experiments. Essentially what I wanted is I wanted to make sure one important thing, perhaps more important than pot size, is making sure that you are trying to keep the distance between your crop and the weeds in this experiment relatively standard. So I didn't want to have one pot where all my weeds were like an inch away and another pot where all my weeds were like, 6 inches away. So that's perhaps more important. Okay, Ruby asks a very good question. In terms of standardizing soil nutrients, how did I do that? So basically, it was by using different ratios of biologically active field soil to sterile potting mix. So in the nutrient plus microbes soil, I was using 50-50 mix. And in the inoculated soil, which is how I tested plant soil feedbacks, I was using a 5 to 95% mix. And this seems like a kind of a coarse method, but there have been methodological studies that have assessed whether or not, one, you can quantify microbial functional effects on plant growth with this ratio, and two, how much of a confounding effect there are about crop nutrients. And the answer is this is a successful and appropriate technique to use. So I hope that helps. So people feel free to unmute yourself too and talk to me, talk to the people. But so Anne asks, can you speak to what kind of farmer may have used practices in the last 4 experiments you presented? Where are your field experiments located? And so the farmers that use my experiment are so this is a forage crop system, and forages are very common in New York and the Northeast. This experiment, the diversity experiment that I was sampling from, and Anne, you managed really well 2016 to 2019 was replicated in New York, Vermont, and New Hampshire. And it attempted to use common forage crops in all three of
Oh,
these states.
Woody, hi.
Hi,
Speak
Ann.
to the people. No, the four experiments that you presented at the end. That's what
Oh, yeah.
I know about my stuff.
Yeah, yeah. I just thought you wanted me to reiterate the fire. So yes, these four experiments are there's always going to be a replication near Ithaca, but we have a large collaboration with the Farm Hub, which is in the Hudson Valley. And so a lot of these experiments are going to be replicated there as well. The cereal rye biomass study has already been collected. That was years ago, and I believe that was here, but I'm not sure. And farmers who could use rotational no-till are farmers who are seeking to reduce, typically organic farmers who are trying to reduce tillage. So say that you're an organic farmer who has terrible tillage issues, you might be interested in using a rolled cover crop system. Also, the rolled cover crop research is, there's also quite a bit of that being done in conventional systems. So it's not out of the question that if I present this work at a farmer meeting, a conventional farmer would also be interested in assessing how different rolled cover crops and cash crops function. So I think that it's, yeah, it could be good. Yes, so Andy brings up a very good point, which is on characterizing the plasticity of functional traits as influenced by the competitive environment. And I agree. Yes, there is when I'm out in the field, I do observe that there is that plasticity. I imagine that this is in some way a follow-up question to my proposal to study weed functional traits by using a database. And the answer to that is that, when doing that project, I would have to be very careful to ensure that I was comparing functional traits which are broad enough that they would continue to apply in different competitive environments. So for instance, just broadly thinking about root structure instead of something as specific as leaf area, which I believe I might have given an example of. So yeah, that's yeah, but that's a great point. And yeah, I think there needs to be some interesting research done in that area. So yeah.
Udi, we are over time now. It's 1:35. So I think great job. I think we can let people go here, people that need to leave. Thanks for attending the seminar today. I believe Udi will stay on for a few minutes and will be available to answer additional questions. But certainly if you need to leave, we understand, and thanks for participating.
This has been a production of Cornell University, on the web at cornell.edu.